Two sires who started the Poll Hereford cattle line that is now commonly used for beef. These are beef cattle, very large. They grow up to about 2,000 pounds as adults, much larger than dairy cows. Poll Hereford line, and there's now carriers for this mutation in Poll Hereford herds, and they've moved from Australia to all over the world, including the U.S. But one of the interesting things about these cows is people would say, well, why would you do a cow for a gene therapy? And it might seem obvious to you that, well, that's a cool thing to do, but it's very difficult to do. It is much harder than treating mice with a genetic disease. There are many obstacles to it. Bringing a sick MSUD cow into the world presented all sorts of challenges and problems. That process alone took us about 3 years working with scientists, veterinarians, geneticists all over the country. It is very expensive to treat a cow. So one of the rate limiting aspects of gene therapies is manufacturing costs. These products are much more costly than making your typical antihypertensive drug or antidepressant drug. And mice are tiny. So you can treat lots of mice with a little bit of material. A newborn calf is 70 pounds, the size of a full-size adult human. And so the manufacturing requirements for the cow are massive. In this case, it cost $1 million to make a single dose in a research environment. And it just, gave me a lot of gray hair. So why bother? What's the point? Well, the cow offers distinct advantages to mice that are actually relevant for what we call translational science, meaning what can we learn from the animal studies that we think we can translate that knowledge directly, map it directly onto the human condition? Because as Dr. Vockley alluded to, one of the biggest challenges in gene therapy development is the simple fact that mice aren't people. Like not even close when it comes to anatomy, physiology, metabolic rate, handling of external substances that are given to them, immune reactions. And so the trail to successful gene therapy is littered with a lot of dead mice. And we can only learn so much. One of the key aspects of MSUD, obviously, is the brain disease. So MSUD is a systemic inborn error of metabolism. We measure the problem in the bloodstream. But the only organ in your body that apparently cares is the brain. It's really the only organ that's affected by these endogenous toxins that accumulate. The mouse brain of MSUD only replicates certain aspects of the human disease and doesn't do so very well, actually. Importantly, mouse brains, although they have the abnormal chemistry of MSUD, they don't get particularly swollen. They don't show a lot of signs of actually the severe swelling that is characteristic of the human disease, which means that whatever's happening in the mouse brain, it doesn't that closely replicate what's happening in the human brain. Not so for the calves. The calves have a neurological phenotype exactly identical to the human disease. Down to every neurochemical, the characteristic massive swelling of the brain, and even the location of the swelling and how water tracks within the brain is essentially identical to what you see in the human disease. So it really gives us a perfect neurological model, which in my view is the most valuable aspect of this model. It's also very difficult to measure what will be one of the most, if not the most important outcome for most humans with MSUD, which is liberalization of protein in the diet. So this is looking at humans. What you're looking at here is a large group of MSUD patients with their age on the bottom axis there. The pink circles represent something called the proportional intact protein equivalent, or PIPE for short. The PIPE is a simple way of expressing what percentage of all the protein in your diet comes from natural intact sources versus medical food sources, where the two sum to 100%. So here you see in MSUD patients with advancing age, by the time they're about 18 months of age, they only tolerate 10% of their protein from natural sources. This is classic MSUD. And so it remains for the rest of their life. Now, one of the key endpoints for a clinical trial, I would say any clinical trial, and this is true of transplant, and this would be true of gene editing, gene therapy, is we want these to reverse, right? We want the red circles to be up on top, preferably at 100%. That would equate to dietary freedom, which is what most MSUD patients want. And this is really hard to model in a mouse. It's really hard to model in a mouse. Because after treatment, you want to be able to give graded amounts. You want to start to very gradually ramp up that PIPE level in the animal and push it as far as you can to hopefully get to 100%. You don't have a lot of options when you buy mouse chow. You have like two options, 20% protein and 40% protein. So you can't, and it's very hard to quantify what mice eat in a day. The cow actually allows you to monitor and gradate these nutritional aspects very carefully in a way you can't do in mice. And then finally, the cow has biomarkers, branched-chain amino acid levels and alloisoleucine levels that are much closer to what humans would have with MSUD. So mice, the classic MSUD mice have like really high leucine levels, often 4,000, 6,000, which is not, we can see those in classic MSUD in humans, but more typically we see levels in the 1,500 to 2,500 range, which is what we see in cows. The other thing that we see in cows that is not as prominent in mice is, for those of you who live with MSUD, you know that one of the things that your dietitians and doctors are constantly doing is they are micro-adjusting your intake of things like isoleucine and valine. And what they're trying to do is they're trying to replicate the concentration ratios of those branch chains with one another. This is actually a really important part of branched-chain amino acid physiology that in MSUD we see these are constantly out of whack. So the isoleucine and leucine are like a seesaw. One goes up, the other one goes down. We see that in the cow in a way we don't see in the mouse. So for all those reasons, we decided, let's try to do the cow. I had learned about the cow in medical school. I first found this 1986 paper buried in the stacks in the medical libraries in the basement. Got my old, photocopier card that I would have to like put in the coins in the coin machine and laid it out and photocopied it. And for some bizarre reason, I saved that article on paper. And I had it sitting in a cardboard box in my office. And I pulled it out and looked at it and dusted it off. And this was around 2018, early 2018. And I called Guangping Gao, man in the bottom left, who incidentally is the person who first discovered AAVs in human tissues. So he's He's one of the real pioneers in AAV, and he led the gene therapy lab at University of Massachusetts, which is a world-renowned gene therapy lab. He and I had become friends and had talked about working on MSUD, and I called him up and I said, Guangping, what would you say if I asked you to make vector for MSUD and treat a cow? And there was a long silence on the other end. And after about a solid 30 seconds of silence, I'm like, are you there? He said, okay. And I said, wow, you took a long time to decide that. Do you think it's a bad idea? He said, no, I think it's a great idea. I was just calculating how much it's going to cost to make the vector to make sure I can afford it. And so this is the team he put together. Shortly after that conversation, I had a medical student working with me at the time. I handed her that old dusty paper and I said, find me one of these cows in the United States. And she did. She found a paper publication of a calf discovered in Indiana on a farm. We called the farmer, who led us to another farmer in Iowa who had delivered 4 or 5 of these animals in his herd. And we took a road trip. I don't know who that guy in the middle is. Looks familiar. It's kind of depressing to see, but anyway. So this is people from the Clinic for Special Children Laboratory, Mark and Lorraine Martin, parents of an MSUD child from the Mennonite community, Don Hardin and his son who own the farm. And over there on the left, poking his head out, is Dan Wong. He is the master engineer who designed the vector at UMass. And he got to come out and stand in cow manure and spin blood samples for the day. We drew blood on about 136 cows that day. We found 4 female carriers, identified a male sire who was a carrier. University of Massachusetts bought those animals and we shipped them to Massachusetts to a farm. So this is the treatment. What they designed is a AAV9. Dr. Vockley mentioned this is not adenovirus. It's adeno-associated virus. It's very different. It's much safer, much smaller. It has the distinct advantage of penetrating multiple tissues. When you give it intravenously, it goes into muscle, it goes into liver, and importantly, it's the only AAV that consistently crosses the blood-brain barrier and gets to nerve cells, which is important for MSUD. One of the things we learned is that these 2 genes, BCKDHA and BCKDHB, which together lock into a complex that mediates the main chemical reaction of the enzyme, are codependent in their tissue expression. What that means is if cells have a broken copy of one of those genes, they don't bother making the other protein because it's a waste of materials and energy. So if one gene is knocked out, both proteins are low, meaning that we could put both genes into a single vector and actually have this vector read in 2 different directions, produce these in equal quantities, and rescue animals, hopefully people, with 2 different types of MSUD, type 1A and type 1B. That doubles the number of treatable patients worldwide. So these 2 each represent about 40% of cases worldwide. This vector captures 80% of the population. This is our patient, Petunia. So our first cow was named Louie, L-E-U-I-E, Louie. We also had a Valerie. They died. These cows are very sick when they're born, very difficult to keep alive. We ultimately lost 3 cows. We managed to keep Petunia alive. She was born in October of 2021, the day before my birthday. You can see her here at 15 hours. She's already semi-comatose. She's got a leucine level of 1,300. We barely kept her alive. We stayed up all night in the barn, giving her high IV dextrose, insulin, lipid solutions, gavage feeding with MSUD formula, sort of side by side with a veterinarian. But ultimately, it worked. We were gradually, day by day, week by week, able to wean up her protein intake, quantify that, and demonstrate that within 70 days, she had achieved dietary freedom and no more crises. This is her current survival curve. So you can see the red line is a mortality curve for all published instances of MSUD cows. There's been 36 cows published in the literature. None of them survived beyond 10 days of age. And median death was about 3 days. They all died in coma with massive brain swelling. This is our little girl. She's coming up on her 5th birthday. She is no longer little. She is now over 1,500 pounds. Biomarkers are stable but not normal. This is not unlike what you can see after liver transplant. So we know from liver transplant that after a successful liver transplant, the branched chain amino acid levels are sort of rock solid stable. But if you compare them to a normal population, they are about 30 to 50% higher than normal values in humans. Not high enough that it appears to have any health consequences, but there's a little trace of MSUD left behind. And that's what we see with the cow. So you can see the leucine levels in blue as compared to the historical cohort of cows. Notice that on the y-axis, the axis is a dual or a broken axis. So you can see there were some cows with levels up as high as 10,000. And the cows hovering typically between about 300 and 750. Most recent levels are 400 leucine level. Importantly, the alloisoleucine has stayed down. Now, alloisoleucine is not something that many people pay attention to. Alloisoleucine is called a pathognomonic biomarker, meaning that it is a chemical in the bloodstream that is unique to people with MSUD. No other creatures on the planet Earth can make this chemical. And so it's the perfect biomarker because it rises and falls in parallel with the level of enzyme activity in the body, inversely, right? So if you have normal enzyme activity, you should have no detectable alloisoleucine. For those of you who are familiar with diabetes, this is more like a hemoglobin A1C. It's like a chronic tracker of the disease state. And you can see we didn't normalize the alloisoleucine, but it came down and it stayed down, which tells us we've effectively reintroduced enzyme activity. Now, something that is interesting about the cow that we couldn't achieve with the human is we can look at durability both across time and durability across growth. So if we want to give these to babies, if we want to take a newborn with MSUD and treat them with gene therapy, a very important question is, what happens to that one-time treatment when that child increases 15-fold in weight by the time they become an adult? Right? You start life at 3.5 kilos, and you're going to get up to an average of 70 kilos. But we're just putting that gene therapy in there one time. Isn't it going to get diluted out? Right? It's like adding, it's like adding water to dye. Like, isn't the color going to fade if you add enough water to it? And this is one of these areas, as Dr. Vockley indicated, we're creating new biology. So the real answer is we don't know. We really don't know what's going to happen. And the cow lets us kind of answer that question. The cow increased 20-fold in weight over a 5-year period, and the therapy's still working. This is something we wouldn't be able to model in a mouse. Does anybody know which one's the MSUD cow? It's the one closest to you. Remember, she has the white marking. So that's Petunia. So she's just hanging out in the field these days with her friends. She's just chilling, eating grass and grain. She seems fine. You can't, we still don't know how to do cognitive tests on cows, so we can't tell you for sure. I think she has a little bit of ADD myself. She seems, they call her Spicy Beast up there. She seems a little bit, she likes to tease the other cows. I just put this up just to tell you that for those of you who are really interested in taking a deep dive, we published all this data in Science Translational Medicine. This is freely available online. So we paid for an open access license to make it publicly available. This is a very technically complicated paper, but if you want to really hear the whole story, it's in there. So one more thing I just want to talk about. This is new data just in from our group, and it was based on a previous paper that was published by Rebecca Ahrens-Nicholas's group, who you heard is on the advisory board here, excellent scientist and physician. They published this really interesting paper where they made two kinds of MSUD knockout mice. If you look on your left, the typical knockout mice, they knocked out one of the genes in the whole body knockout. So they had a classic MSUD mouse. And when they measured the blood, all the biomarkers were elevated. When they measured the brain, all the biomarkers were elevated. They then made a mouse through some very clever chemistry that only had the gene knocked out in the brain, but not the rest of the body. So, remarkably, all the biomarkers were normal in the blood, but they were not normal in the brain, which means that if you have perfect dietary control of your MSUD, or you have a liver transplant for your MSUD, it will make you a much healthier person. You'll have a much better quality of life, but all is not well in your brain, actually. There's a neurochemical disturbance, a number of neurochemical disturbances in the brain that persist even under the best therapies. So what we did is we took that observation, we did essentially a mirror experiment where we took our mice with MSUD, we drew blood and we took homogenized brain. We sacrificed the mice, we took samples simultaneously. And then we subjected them to a large chemical panel, measurement panel. And what we found is if you look at the mouse who's not treated, nothing surprising here. 2-ketoisocaproic acid, which is the real potent metabolic poison in MSUD. It's the keto acid derivative of leucine. It's probably the most potent toxin in MSUD. It's about 9-fold elevated in the brain and about 100-fold elevated in blood. 2-ketoglutaric acid is an energy metabolite that gets elevated in response to the presence of ketoisocaproic acid. That's interesting chemistry, which I can talk about offline if anybody's interested. But importantly, remember we talked about glutamate as the neurotransmitter in the early talk, as being the neurotransmitter for 85% of neuronal synapses and the main driver on brain energy metabolism. It's depleted 50% in brain of the animals. So then we gave them our vector. And this is fascinating. So what we found is after treating with the vector, 140 days later, we looked at the blood and brain, and we found that it completely corrected brain chemistry, but not blood chemistry. So the 2-KIC level was still 4-fold elevated in blood, but the mice were asymptomatic. What this means is the gene therapy penetrates the brain, reintroduces enzyme activity in the brain, and actually protects the brain from the circulating neurotoxin. So it provides a safety net against the blood metabolites. And that was a finding that really sets gene therapy apart from other available therapies. So I just want to thank all of my collaborators. This is really an amazing group of people. This is project is 8 years in running now. We have some additional experiments planned, which I hope I can talk to you about in the next year or two. So thanks, everybody.